G'day guys. Today's a sort of a video that's going to cover some other aspects of animal-based nutrition, and in relation to cancer. We hear a lot of things that are told out there, in terms of epidemiology. But as we all know, epidemiology can never prove causality of any sort. It's in many cases, even processed meat is like 1.17, like 17%. It's irrelevant. It doesn't even get to 2 or 3 in terms of epidemiology, observational epidemiology, to even warrant, something like a clinical experimental study, to generate a hypothesis, so to speak. And most epidemiologists recognize that you need at least to have an odds ratio of 6 to 10. And when you're looking at any complex sort of factors, higher than 10. So when you're looking at basically even cancer, the rates were an odds ratio of 10 to 30, 1 point something like that. And that's pretty much irrelevant. And even of those very high odds ratios of 10 to 30, we still know there are indigenous populations that consumed, that were on an animal-based diet, like the North American Plains Indians, the Inuit and others that consumed moderate, not a lot of tobacco, but did consume some tobacco and had no history of cancer, cardiovascular disease or anything. So something in the diet must have been protecting them, so that is a confounding variable that was at play, so to speak, and that's why epidemiology can never prove causality. It only can infer that there may be something in here, and the stronger the inference, obviously, the stronger the probability that something is likely. But that still needs to be in the very high levels. When you've got 1 point something like that, it's laughable to be, it's beyond laughable in some regard. Now, let's just go through some of the backgrounds in terms of certain pathways, which are very important. And then we can look at some of the other aspects in that regard. So I'll just share my screen. In this regard, there'll be 2 areas that I'll be looking at. So, okay, there's this area. So we're basically looking at the role of PTEN signaling in terms of cancer. This is in the US, this study was done. Okay, so that's a review paper looking at some of what the literature has actually been looking at, saying what they're finding a very strong relationship. So they go, PTEN signal tumor suppressor was discovered by its homozygous deletion in other mutations in cancer. So homozygous, basically there were studies where they actually deleted the specific genes to basically see what would happen and stuff like that. That's what they're talking about in the sort of past. So in studies. So the PTEN has been shown to be a non-redundant evolutionary conserved phosphatase whose function affects diverse cellular processes, such as cell cycle progression. Now, cell cycle progression, just so people to understand, that's just the normal, your cells normally, they will basically divide and they'll proliferate, but it's all controlled, it's highly regulated. It's just that your normal cells turning over. So all those mechanisms that are actually that's what they mean by that in that regard. So where was I again? Cell proliferation. Well, that I don't need to explain that. That just cell proliferation. That can be normal cell proliferation in tissue. If you're doing really a lot of pumping iron and stuff like that, you're going to basically be building muscle much faster. You're going to proliferating muscle much faster, tissue. That's normal. But it also could mean, cancerous type of proliferation, all sorts of stuff. Chemotaxis. I think I've pronounced it. I hope I've pronounced it correctly. That's the sort of how can I put it? it's sort of like, you know, just think of taxi. It's sort of the cell movement, in a sense. That's how I remember it. I just remember it from taxi, taxi, sort of thing. I should move my camera, but anyway, it doesn't matter. I'll keep on looking back and forward. Apoptosis, cell death. Aging, well, it's just the normal cellular aging. Your cells go through different stages. They age, they become slightly more, depending on your exposure to different things and all that, they can become a bit senescent. So muscle contractility is basically, well, it's just, the filaments. Remember, Bart talks about the filaments, how they contract with force. That's basically what it's talking about here. It's basically, this also affects that muscle contracting, sort of the force of pulling inwards those filaments and that sort of stuff. That's basically all that's talking about. DNA damage response. There's signaling pathways in the body that basically sort of, they detect certain changes or damages, and they sort of have a certain type of way of responding, signaling and saying to the body, well, we've got a bit of a problem here, we need to sort of fix this. I mean, when you're sleeping at night, well, if you're following your circadian rhythms and being a good boy and girl, then you should basically have the right signaling, and that melatonin should basically be helping with that as well. Cell polarity, that's a bit of a, to get your head around that sort of thing, is, oh, hold on, I missed angiogenesis. That's just blood vessels. So that's all, when you think about, angiogenesis, that's just blood vessels being grown and stuff like that, new blood vessels. But polarity is actually the actual shape, sort of, how can I put it? All cells have a certain shape, or, a certain structure and a certain shape, that they function in a certain way, and they, sort of what it's called, and they may slightly differ and all that. One thing we know about this sort of thing, so this pathway plays an important role in this, and we do know that even in early cancer, the shape of would, it will lose, the cancer cell will lose its sort of it's what they call polarity, but it's sort of, it's the cells, the way they are sort of pulled together and stuff like that really changes. They lose that sort of structure, that normal cell structure, and they can sort of, I think they use it basically to sort of invade into adjoining tissue and sort of proliferate in metastasis. But even in early stages, they talk about this sort of, it's sort of the cell membrane, the sort of, the way cells adhere to each other. When the first cell division that sort of happens in the body, there's like controller cells, and they can sort of control the different, and they make sure that they sort of adhere to each other, and so they work as a group in a sense. Sort of, cancer is basically when they're no longer a group. They're like independent. Well, I mean, I'm going independent, mate. I'm going to do whatever I like in the body. So it's a bit like that, and sort of that's what they're sort of talking about when they talk about polarity. I never managed to describe that very well. It's one of those things that, but it's, anyway, let's move on before it becomes too long, this video. In accordance with the ability to influence multiple crucial cellular processes, PTEN has a major role in pathogenesis of numerous diseases like as diabetes, autism, and almost every cancer examined. So every single cancer that they've examined, PTEN is basically heavily involved in signaling is modified in cancer, and how these changes correlate with and might possibly affect the action of targeted chemotherapy. So they're looking at basically what sort of medications that can they create and use in targeted chemotherapy. Well, let's see what really can actually, and there is another pathway. I need to get to Wikipedia for that. And it's this one here, basically. This is an enzyme called caspase-9 enzyme that in the human is encoded blah, blah, blah for that gene. It initiates caspase critical to apoptotic pathway, which is again cell death, found in many tissues. So this caspase-9 homologs have been identified in mammals, for which they are also known to exist, such as mackerel, blah, 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 blah. So it's a mammalian enzyme, basically. Humans and other mammals that basically have this, and it's involved in this sort of apoptotic activity. So I can get rid of that and continue with the other stuff, the real stuff, the crux of this video. Anyway, our dear friend taurine. Yes, taurine attenuates, which is reduces carcinogenicity in ulcerative colitis, colorectal cancer mouse model. This is a Japanese study, so another mammal, and these pathways, both these pathways are in, as you can see, that one and that one. Basically, they are both in the human and mammalian tissue. So this mouse model, when we're looking at, we could use this. Taurine is a type of amino acid that numerous physiological and therapeutic functions, including anti-inflammation. I've talked about that in the past. However, there are a few studies on the anti-cancer action of taurine. Our previous studies have demonstrated that taurine exhibits an apoptosis-inducing effect on human nanophalangeal carcinoma cells in vitro. In this study, we have investigated whether taurine has an anti-cancer effect using, so they're using this sort of azimethane slash sulfur sodium. So it's a preparation to induce cancer. So it's like a cancer agent, induced mouse model for colon carcinogenesis. So they're using this to basically induce cancer in this mammalian model in that way. All mice, except those in the control, received a single. So they've got a control, which is really good. They've got basically one group that is basically not getting any of the so they've got a control with that they haven't put anything in, that they haven't put this cancer producing substance. They've got basically the other two that are both have been given their cancer producing substance, but one is being supplemented with taurine, the other isn't. Okay, so we're quite clear on that. And except for the control, received a single intrapreneural injection of so only those two got that product in the drinking water for 7 days twice and with 1 week interval. After the first DSS treatment, mice were given distilled water model or taurine in the drinking water of the taurine group. So the ones that were given were induced cancer. One group was only given the distilled, and the other one was given the taurine ad libitum, so they could have as much as they liked. No tumors were observed in the control group, which is exactly what we would think, because they weren't given the substance to induce those things. Taurine significantly suppressed AOM plus DSS induced tumor formations. So it basically significantly suppressed the formations of these things. Histopathology, basically, where you're looking at the actual tissue itself, they kill the animal, they look at the tissue and see exactly, what it looks like, what the cells look like, how damaged, how deranged is the colon of these animals. So we'll show those shortly. Examination revealed that this cancer type treatment induced colon cancer in all mice, 100%. So all mice had, so we know that basically they did have cancer, all of them. And taurine significantly inhibited the progression of colon cancer by 44.4%. Taurine significantly attenuated, which is reduced cell proliferation in cancer tissue, detected by Ki-67 staining, which is a method that they use to basically look at that tissue. And I'll be showing that shortly. Taurine significantly increased the level of apoptosis, which is cell death marker, which is our dear friend, the Cas9 one that I talked about earlier, that I showed from the Wikipedia, and the tumor suppressor protein, PTEN. Okay, so we've got both these things that are basically been shown, and I've shown you that basically they have a direct relationship to all these processes in the cells. We're talking about the cycles, normal cycles that cell go through. Just stop that so I can just talk to you guys directly. So the normal cycles that cell go through, their normal aging, their normal sort of their movements in the body. They're basically inducing, if they're problematic and have problems, senescence or whatever. Remember I talked about the T cells, how they are protected from becoming senescent because of taurine that plays that role in the thymus. Well, again, even here with all cells in the body, it has basically this protective mechanism in terms of senescence as well. So, and also other things that I've talked about in that regard. So the damaging, the signaling, that pathway that actually plays that role, critical. Taurine again interacts with that pathway, has a direct effect on that pathway, as this study shows. So it's interacting at that level. These are the two key apoptotic pathways, but we know that the PTEN pathway also plays significant roles throughout the body. This is what I've been saying for a long time, that taurine, which is a key sulfur amino acid found primarily only in animal foods, plays a critical role for the normal functioning. This is why traditional populations like the Inuit, like the Maasai and all that, we don't see cancer in those populations. We don't see a lot of the diseases, as we call them, of civilization, because the body is properly regulated with a species appropriate diet. And that's critical. Now, when we get to cancer, there's other things. Here we can use basically higher doses of certain things and do interventions in a certain way. So let me just reshare my screen to continue so we can provide some additional evidence of this study. Anyway, so that's that part. I'm not going to go through this because it'll take too long. That's the general information which I've already described about the experimental model design. That's the length of time and stuff like that, so people can go away and actually look at it themselves if they're interested. This is the sort of length of time that they put them through and all that sort of stuff, which is interesting. Then their fecal blood score, as you can see, they didn't include the control here because it's irrelevant. So as you can see, the model basically has fecal blood damage. So taurine, because it's reducing the effects of the cancer, it's reducing also the fecal blood. So there's quite clear, there's a difference between the two. Now this is the actual colon weight Obviously, if you've got proliferation and cancer cells, you're going to have slightly more weight. Obviously, compared to the model, which is the one that didn't get the taurine, is going to be higher weight here. But when we're looking at numbers of tumors, so here's an important thing. Look at the dramatic difference. There's a control, obviously, nothing. And when we're looking at here, the number of tumors are significantly lower. So you still may have things like non-benign growth that actually happens in that regard, where there's derangements in the cell of the, those neighboring cells and all that. You can actually have senescence and derangement and stuff like that. But it's actually suppressing, attenuating, suppressing, reducing down the number of tumor cells that are actually, where in the model is just proliferating, out of control. Taurine is really holding back and suppressing that ability, slowing it down quite a bit in that regard. So very powerful stuff, especially if you're in the early stages of cancer and stuff like that. The thing that you want to do the most is slow down the progression of the disease itself. There it is. You're actually seeing it in this animal model, significant slowdown in terms of the number of tumors. That is really important in that regard, guys. So when we're looking at basically these, so these are the stains, where they'll slice and create these stains, as they call them, H&E staining. We can actually see that's the normal in the control, obviously. There's no problems. And you can see here's the model, inflammatory polyps. This is the one that actually far less because of the taurine. When we look at the colon cancer tissue, see there compared to the normal, that first part of the slide, that's further zoomed in. Even zoomed in, you're actually seeing, yes, there is still some cancer there and all that, but it's been suppressed. Where here, that's the normal. So that isn't as damaged, but look at the damage in the model. Severe damage, severe derangements of the colon, severe derangements. Taurine from animal foods is suppressing it. This is why we see improvements in basically the functioning of the colon. I've talked about, the whole gastrointestinal system, how the stomach is improved by the acidity levels, but also about the gut motility. That means the contractile, the actual muscles, the actual gut, how they contract and all that, the motility, how that is regulated with taurine and improves the function, but also the protective effect. There's the other aspect in terms of taurine, in terms of cell death within this tissue to get rid of the stuff that should get rid of and stuff like that. So this is really important stuff. As you can see, the number of colon cancers, very low in comparison, less than here. We've got nearly, that's from the control, got 1, 2, 2.5 times, where here you've got something like 0.7 or whatever. So, so basically we're talking about like a practically 3-fold difference in the number of colon cancer, in that regard. So that's a significant difference, especially, if you're looking for survival purposes. So here we're looking at basically at cancer cells and cell polyps, so this is that KI67, just so we can remember that, what they detected in the staining on the proliferation. So you can look at the that's the normal, that's the taurine one. Here's the model. Look at the derangement here, massive derangement. Here there are issues, but there isn't massive derangement either. Again, the normal in comparison, far more derangements, And so here's also the score, the PTEN score. Cancer cells in polyps, look at the taurine levels. So that with the taurine, getting far higher score of PTEN. That's what you want. You don't want lower, you want higher. That means you want that to be expressed much higher to basically, because that is protective, you know. They want to create in the other study, remember we were the previous one I was looking at, they say, oh, let's create a drug that actually increases this. Well, you don't need a drug, just need taurine. Simple. Eat a steak, you'll increase this PTEN score, you know. And you don't have to worry about, you know, the mTOR stuff and all that, the growth pathways. With a 1.3 of insulin to glucagon, the growth pathways are quite low, just enough for muscle protein synthesis. There's not enough signaling there. There's low signaling, you know. Leucine only pushes it up to a certain level of signaling. We've seen it even in a fasted state where you've got really suppressed insulin and stuff, very poor IGF-1 signal. You're still getting muscle protein synthesis because it's been driven by leucine. People should check out my leucine video and understand about that. They should not concern themselves about those growth pathways because they, in a low carbohydrate, ketogenic, in this sort of state, you're not getting those magnified effects. You're only getting primarily signaling for muscle protein synthesis, not these aberrant conditions. And with this sort of stuff, when you throw taurine in, you're actually affecting the signaling of the way these things are, the way cells move, the way they function, the apoptosis of cell, forcing them into death, you know, where you want these numbers to go up. And only taurine from animal foods actually pushes them up. Obviously, somebody who on a species appropriate diet will have that protection automatically. Somebody who's in a cancer state, which I'll discuss at the end of this video, there they have to biohack and do slightly different levels of taurine. So we'll get into that shortly, but it's just to show quite clearly what we're looking at here. Then we're looking at basically, oops, we've covered that. Now let's go to the next one, which is basically the cleaved Caspase 9. Again, as you can see, more expression of this apoptotic within basically the taurine group. Again, quite clear, you know. And then they make their conclusions. This first study has demonstrated that taurine significantly reduced carcinogenicity in vivo, which is important within an animal model in this case. So it's not in vitro because all the other stuff had been in vitro in petri dishes. This is actually a living organism, which is important, which has got very similar pathways like all other mammals. So I would say that it has some applicability to us. We can read something into that. Unfortunately, we can't do experiments on humans to test this theory, and you know we should be doing, but unfortunately, you know, they're basically you would never pass it through an ethics committee because ethics committees say you can't use taurine to basically do experiments to determine whether somebody will have progression of cancer or not. You've got to basically intervene with chemotherapy and other things. And so, yeah, you never get it past an ethics committee to put one group to allow them for their cancer to progress and another group to maybe, you know, in their opinion, not, you know, so it'd be considered unethical. So we can't do that. We have to do the animal models. It's just the reality of it. But I would say since those two pathways that are basically conserved within our, remember, you know, conserved evolutionarily, you know, oops, that's what I meant, evolutionarily conserved in all mammals. I think we can basically quite clearly from that know that it's going to work that taurine will have a similar effect in all mammals because we've seen it in other models as well. It is suggested that taurine attenuates, reduces cell proliferation and induces apoptosis, cell death via the PTEN induction. critical pathway conserved in our genetics of all mammals. Taurine could contribute to the suppression of inflammation-related carcinogenesis. Pretty much. And these Japanese researchers have they basically have no conflict of interest in this regard, obviously. And also, this is the nice thing about these researchers. They have said that all data are available on request to the corresponding author. So they will provide you all the experimental data. We know a lot of other researchers don't. They make conclusions and assumptions, but they don't provide the data. They keep you in the dark. They say, oh, well, this is what we found, but they won't give you all the data collected. Very important. I like it when researchers actually say, we're going to give you the data. If you want to drill through it and check to make sure that we did the right thing, we'll give you the data. I like that. So that gives me confidence in that sort of stuff, that they are basically completely open and candid, that they will share their data with anyone. So if anyone wants that, there is information at the bottom there that actually provides the parties supporting and all that, the sort of the numbers, and you could probably Google and find out the researchers and basically send them a letter or an email to their institution and ask them to provide that information if you're interested in going into it in any deeper way. But I think the take-home message here is the critical importance of taurine in the diet for that general protection, proper regulation of our cells for us healthy people, so we never have these sort of problems and all that in the future. Obviously, if you're not eating a species-appropriate diet and you're magnifying mTOR, IGF-1 and insulin to very high levels by Randall cycle activation, by eating very inappropriate, combination of very refined oils and sugar, well, that even supplementing taurine is not going to really help you. Let's be honest about this. This reduces, it doesn't eliminate. This is just another component you can throw in there to basically keep you healthy. And if you're down, and if you're suffering with cancer, I've already discussed in my previous videos about the chemoprotective effect of taurine, very important. But I would basically, and also there's one proviso, always check, I'll put the video there, check the one about, the side effects the interactions with certain medication that can happen with taurine, and that you need to adjust them with a physician in that regard. But I'll put that somewhere there in that regard, so people can go and check that like that, so they can understand that if you're taking taurine in high supplementation and you're on specific meds, you need to basically, have adjustments made, and you've got cancer at the same time. So let's go back dosing. And this is not a medical advice at all, but if you eat massive amounts, like a kilo of oysters, you will get 10 grams of taurine. Now, obviously, I wouldn't encourage people to be eating, because it'd be very expensive to eat that amount, but it's not going to affect you. Taurine, we've seen in massive doses, very higher, and we haven't seen any real effects. And only those massive doses, there are some people, small percentage of people, that excess sulfur can be a problem, but it's usually if they're on a veganerized or non-animal-based diet. On an animal-based diet, you're getting choline, which will methylate away any excess, and you're getting a lot of methionine, which gets converted to choline through enzymatic pathways and can also methylate away. So non-issue for carnivores or low carbers and all that in terms of dosage, high dosage. But I would basically work my way up, because it is an osmolyte, so it does regulate the electrolytes as well. So you get the bonus of that. But at the same time, you've got to go slow. So you've got to start at 4 grams for a couple of days. I would say a week, so 2 and 2 in doses. Then the next week, go to 6, 3, 2, 2, 2, and then increase that to 3, 3, 3. So you get up to 9, and then one of those, you can pick the middle of the day as 4, and then it's basically 3, 4, 3. You can go straight up to 10 from basically from the 6 in that regard, but you'll move up that. Just keep an eye on how you feel and stuff like that. If you're, I'm not feeling, it's sort of in a way, get a choline supplement and use a bit of choline in that, or eat a couple of eggs. The easiest way is just eat a couple of eggs every day. You get the extra cholesterol, you get a lot of the other phospholipids, which are really important for your body. So just eat a couple of eggs, and that should sort things out pretty fast in that regard. But this is my personal opinion. This is not medical advice. If I was in basically a state of cancer and all that, I would go to these doses. This is not for the general public or general people that don't have cancer. If you don't have cancer, you don't need to go to this level at 4 grams at the most, which is sort of, helps with, our current C19 issues, which I've talked about in other videos. So for those that want to basically be more protective in terms of T lymphocyte, stuff like that, protection, your good friend, taurine will help you in that regard. But for people who have got active cancers, it's really important to basically get the levels right up there to basically maximize apoptosis. We want to knock these cancer cells for 6. That's the focus and all that. And we want to sort of try and regulate cellular functioning much better. It's really important, even, because cells are basically senescent, really severely damaged, or the proliferation in metastasis is actually happening and other cells are being damaged quite a bit. We don't want them turning cancerous. We want apoptosis, to maximize apoptosis and kill them off. And remember, even fasting in a metastatic state can actually be protective of cancer cells, not only normal cells. So that's a problem. You can't use fasting as a way of apoptosis anymore in a metastatic state. In early stages, you can sort of use it in that regard, but at later stages, it can actually work against you. We want to use the substance where it doesn't matter what stage you are in the cancer life cycle. We want to basically be able to apoptotically knock it off at this stage, that stage, that stage, and that stage, metastasis. So we basically want to create hostility in the body and apoptotic cell death maximize that as much as possible within somebody with cancer. Now, if you want to go down the chemo route, taurine has been shown in high doses, again, to be chemoprotective. I've shown that research before, so in one of my previous cancer videos. So that's basically it, guys. So I hope you got something out of it for maybe yourself or a rally, that this information could be quite valuable. Remember, we need to protect, even if people take the route of chemotherapy, we need to protect the normal cells. And that's where taurine plays a big role. We've seen the chemoprotective, and I've discussed the T cells when they actually fight off viruses, whatever, they get to sustain certain damage. Well, taurine comes in and helps them in that, in reducing the amount of senescent damage that they sustain. Very important. So same stuff, protecting normal cells from chemotherapy. So really important, because we know that when you damage the cells too much, even you may go into remission, but when it comes back with a vengeance. So that's what research has shown. So I'll leave people up to their, it's not for me. You need to work with an oncologist and, try these high doses and keep yourself monitored. And at the end of the day, you need to take responsibility for your own life and determine which course of action you take. It's not for me to tell you. All I'm doing here is providing you with some information on the usages of taurine in cancer based on this research that is, this first type of research in vivo that actually looked within an animal, a mammalian animal model, which has the same sort of pathways. These are conserved genetic pathway, that actually work in a certain way. And we can actually basically be confident that, if it has in this, that it should have in us as well. But how much, there are differences between organisms in terms of signaling and stuff like that. So we don't know. But you know what? If you're in that state, I'd throw everything in the kitchen sink if I was in that condition. But at the end of the day, that is a choice for you to make. Anyway, see yous.